Hello all of you. Today I would like to discuss about C4 pathway or C4 cycle or Hatch and Slack pathway, and after that I will talk about cam plants, and in the end I will talk about the external and internal factors which affect the rate of photosynthesis in plants. So let's first start with C4 pathway of CO2 fixation. So for considerable period of time, the Calvin cycle as described earlier was thought to be the only photosynthetic reaction sequence operating in higher plants and algae. But in 1965, three scientists whose name was Korchak, Hart and Burr reported that four carbon containing dicarboxylic acids that, that were malate, aspartate, were the major label products when sugarcane leaves were allowed to photosynthesize for short periods in carbon 14 labeled carbon dioxide. This finding was confirmed greatly, and further it was elaborated by Hatch and Slack in the year of 1970, and who observed a heavy leveling of oxaloacetic acid, malic acid and aspartic acid when sugarcane leaves were exposed to carbon 14 level carbon dioxide for 1 second. So this and further studies by these workers led to the establishment of yet another CO2 reduction pathway, which is called as Hatch and Slack pathway. And because the C4 dicarboxylic acids are the earliest product, so this pathway is also known as C4 dicarboxylic acid pathway. So since in this pathway the first stable product is 4 carbon containing compound, that is oxaloacetic acid, that's why this pathway is also known as C4 pathway. And in this pathway the initial carbon fixation takes place in mesophyll cell and the Calvin cycle takes place in bundle sheath cell. So here the carbon fixation takes place twice here, one in mesophyll cell and another in bundle sheath cells. In this pathway, phosphoenol pyruvate carboxylase attaches an incoming carbon dioxide molecule to the 3 carbon molecule, that is phosphoenol pyruvate, which ultimately leads to the production or synthesis of oxaloacetic acid, which is a 4 carbon molecule. This pathway has been found to operate in many plant species of the family Gramineae, and the examples are sugarcane, maize, sorghum, etc., which are grouped together as tropical grasses. And C4 plants are distinguished from other plants by two ways. First is C4 plants generally don't have photorespiration process, right? So that's why they are considered as photosynthetically efficient plants, right? And second is that all C4 plants show similarity in the anatomy of their leaves. So now I would like to discuss about the leaf anatomy of the C4 plant, and by this I will also try to differentiate the leaf anatomy of C3 plants and C4 plants. So this is the leaf anatomy of C3 plant. This is the leaf anatomy of C4 plants. So here just notice this ring shaped cell, and this ring shaped cell in both C3 plants and C4 plants are known as bundle sheath cells, and the short and the smaller cells which enclose this bundle sheath, or in small cells in enclosing these bundle sheath cells, are known as mesophyll cells. So now I would like to discuss about the main differences in the uh, leaf anatomy between C3 plants and C4 plants. So the first different So the first important difference is in C3 plant, the bundle sheath cells are not compactly covered by uh, mesophyll cells, right? So you can see here gap is present here, right? But in C4 plant, the bundle sheath cells have been compactly enclosed by mesophyll cells. Second difference is that in C3 plants, the bundle sheath cells don't have any chloroplast. That's why they are looking transparent or white in color. But in C4 plants, the bundle sheath cells have chloroplast in it, and that's why they are looking green in color. Third difference is that in C3 plants, normal chloroplasts are generally present, which means grana are generally present in the chloroplast present in C3 plant. But in C4 plant, dimorphic type of chloroplast generally present. Here dimorphic means two kinds of chloroplasts are generally present. First kind of chloroplast presents in mesophyll cell, which generally have grana in it. But the second kind of chloroplast generally present in bundle sheath cells, which generally don't have any grana. So by this reason, the chloroplasts which are present in C4 plants are of dimorphic type, right? So the leaf anatomy of C4 plants of are generally known as Cranch anatomy, and why they are known as Cranch anatomy, because here you can notice the presence of radially arranged cells of bundle sheaths around their vascular bundles, which looks like ring or wreath, right? And in German word, you can in German word Kranz meaning wreath. So that's why the leaf anatomy of C4 plant is known as Kranz anatomy. In C3 plant, the bundle sheath cell do not contain chloroplast. In C4 plants, the bundle sheath cell contain chloroplast. I already discussed about it. In C3 plants, the carbon dioxide fixation takes place only at one place, and in C4 plant, carbon dioxide fixation takes place twice. So since in C3 plants, chloroplast present only in mesophyll cells, that's why the carbon dioxide fixation takes place only in mesophyll cell in C3 plants. But in C4 plants, chloroplast present at both mesophyll cells and the bundle sheath cells. That's why here carbon dioxide fixation also takes place in both these cells, that is in mesophyll cells and bundle sheath cells. Now I would like to discuss about the C4 pathway or Hatch-Slack pathway in detail. So here in C4 pathway, the CO2 fixation takes place twice. First CO2 fixation takes place in mesophyll cell, and second CO2 fixation takes place in bundle sheath cell. So now at the initial stage, atmospheric CO2 enters into the mesophyll cells, and it is generally accepted by phosphoenol pyruvate, and with the help of enzyme, that is phosphoenol pyruvate carboxylase, phosphoenol pyruvate converts into oxaloacetic acid, which is a 4 carbon containing dicarboxylic acid. And this oxaloacetic acid is now reduced to malic acid or malate at the expense of one molecule of NADPH with the help of enzyme, that is malate dehydrogenase. And this malate then diffuse into bundle sheath cells, and after after it's entering into bundle seed cells, its decarboxylation takes place, and as a result of that, this malate is split into two molecules, that is one molecule of CO2 and one molecule of pyruvate with the help of enzyme malic enzyme. And here CO2 enters into the Calvin cycle, which ultimately leads to the biosynthesis of glucose. But here pyruvate again enters into bundle seed cells, and at the expense of two molecule of ATP and in the presence of enzyme pyruvate phosphate dikinase, it again converts into phosphoenol pyruvate. So by this way, phosphoenol pyruvate has been recycled back. So this is all about the C4 cycle, right? Now here question comes that how many total ATP and NADPH are required for the fixation of one molecule of CO2. So from Calvin cycle, you know that 3 ATP and 2 NADPH are required for the fixation of one molecule of CO2. And here the fixation fixation of CO2 takes place twice, right? So in mesophyll cell also two molecule of ATP are required for the conversion of pyruvate to phosphoenol pyruvate. So you will have to add this 2 ATP here, and then you will get the total, you will get an idea about the total ATP and NADPH required for the fixation of one molecule of CO2, and that is 5 ATP and 2 NADPH are required for the synthesis of, or you can say for the fixation of one molecule of CO2. Now another question comes that how many total ATP and NADPH are required for the synthesis of one molecule of glucose in C4 cycles. So as you know that 6 cycles are required for the synthesis of one glucose molecule. So you will have to multiply this figure by 6, and when you will multiply it, then you will get an idea about the total ATP and NADPH required for the synthesis of one glucose molecule, and that are 30 ATP and 12 NADPH are required for the synthesis of one molecule of glucose in C4 cycle, right? So this is all about the Hatch and Slack pathway or C4 cycle. Now I would like to talk about the differences between C3 and C4 plants. So in C3 plant, the carbon dioxide fixation generally takes place in mesophyll cell, but in C4 plant, carbon dioxide fixation takes place twice, first in mesophyll cell and second in bundle sheath cells. Another important difference is that first stable products of C3 plant is 3 carbon compound, that is phosphoglyceric acid, and the first stable product of C4 plant is 4 carbon containing compound, that is oxaloacetic acid. And another main difference is C3 plants are less efficient due to the higher rate of photosynthesis, but C4 plants are more efficient due to less photorespiration, right? In C3 plants, total 18 ATPs are required for synthesis of one molecule of glucose, but in C4 plants, total 30 ATPs are required for the synthesis of one glucose molecule. And C3 plants are, examples of C3 plants are paddy, wheat, potato, and so on. And examples of C4 plants are sugarcane, maize, sorghum, amaranthus, and so on. So these are the main differences between C3 and C4 plants. So here one question can come into your exam that elaborate the differences or write the differences between C3 and C4 plants. Now I would like to discuss about the crassulacean acid metabolism, or it can also be known as dark CO2 fixation. So in most plants, the stomata, which are like tiny mouths that take oxygen all along the surface of their leaves open during the day to take carbon dioxide and release O2. Most plants open their stomata during the day because that is when energy is received from the sun. The energy from the sun is harvested by the chloroplast and used to make ATP and NADPH. These short-term energy storage molecules are then used to power the fixation of carbon into sugar. But in plants living in very dry environments, however, dangerous amount of uh, water can be lost if stomata open during the hot dry days. And during the night, which tends to much cooler in dry environments, far less water is lost by opening the stomata. So now, in order to meet their needs to combine the sun's energy with carbon dioxide from the air, CAM plants take in carbon dioxide at night and store in the form of 4 carbon acid called malate. Then malate is released during the day where it can be combined with ATP and NADPH created by sun's energy. It is also known as assimilatory power, right? Which can be, which are generally generated in light reaction. The name crassulaceous acid metabolism comes from crassula plant, which was the first place that CAM metabolism was discovered and studied. Now I would like to discuss about the CAM pathway or dark CO2 fixation in detail. So in the night time, stomata opens in CAM plants, right? And through this open stomata, CO2 enters into the cytoplasm, and here in the cytoplasm CO2 react with H2O in the presence of enzyme carbonic anhydride, which ultimately leads to the development or synthesis of bicarbonate. And this bicarbonate are generally accepted by 3 carbon compound, that is phosphoenol pyruvate, and in the presence of enzyme, that is phosphoenol pyruvate carboxylate, it converts into oxaloacetic acid. Now this oxaloacetic acid is reduced to malate uh, uh, by the expense of 1 NADH and in the presence of enzyme malate dehydrogenase, and this malate then enters into or diffuse into vacuoles where it remains here in accumulated form. So here the fixed CO2 in CAM plants remains in vacuole in the form of malate, and since malate is acidic in nature, that's why vacuole here become acidified organ. Now when day comes, this stored or accumulated malate again diffuse into the chloroplast, where it converts into oxaloacetate in the presence of enzyme malate dehydrogenase, and this oxaloacetic acetate again converts into 3 carbon compound, that is phosphoenol pyruvate in the presence of enzyme phosphoenol pyruvate carboxykinase at the expense of 1 molecule of ATP. Actually here decarboxylation of oxaloacetic decarboxylation and phosphorylation both are taking place for oxaloacetic acid. So you can say that ATP has been utilized here. Now oxaloacetic acid has been converted into 4 carbon compound, converted into 3 carbon compound, that is phosphoenol pyruvate by both phosphorylation and by both of its phosphorylation and decarboxylation. So the carbon dioxide which has been liberated from oxaloacetic acid now enters into the Calvin cycle, and through Calvin cycle it forms glucose or carbohydrates. So by this way In camp plants, carbon dioxide fixation takes place in the night, and and synthesis of carbohydrate generally takes place in the day. Right. So this is all about the CAM plants or CAM pathway. So now I would like to discuss about the uh, difference in the characteristic between C3 plants, C4 plants and CAM plants. So on the basis of the distribution in the plant kingdom, C3 plants, 85% of the plants are C3 plants, 3% are C4 plants and 8% are CAM plants. In C3 plants, carbon dioxide fixation takes place by C3 cycle. In C4 plants, the similar process takes place by both C3 and C4 cycles. And in CAM plants, again, the CO2 fixation takes place by both C3 and C4 cycles. So the main difference between C3, C4 and CAM plants is based on their stomatal movement. So in C3 plants, stomata opens C3 and in both C3 and C4 plants, stomata open at daytime and close at night. But in CAM plants, stomata open at night and close in the day. And C3 plants are generally present in temperate region. C4 plants are generally present in tropical or semi-tropical region, and CAM plants are generally found in desert or arid habitats. So the question can also come from this slide that elaborate the characteristic difference between C3, C4 and CAM plants. Now I would like to discuss about the factors affecting the photosynthesis. So here both external and internal factor affects the rate of photosynthesis. So the most important example of the external factor is light, and it affects the rate of photosynthesis in three ways. So the here light quality, light intensity and duration of light, all the three put effect on the rate of photosynthesis. So the rate of photosynthesis is higher in white light than in monochromatic light, that is light with rays of particular color or wavelength. So another way is the rate of photosynthesis increases with increasing light intensity, but extremely intense light may have direct inhibitory effect on photosynthesis, and this phenomenon is known as solarization. The rate of photosynthesis is greater in intermittent than in continuous light. So why it is so? So just go to my previous class and just read the red drop and immersion enhancement effect, and then you will come to know about the reason why rate of photosynthesis is greater in intermittent light than in continuous light. Another external factor is concentration of CO2 in atmosphere. So an increase in concentration about 1% increases the rate of photosynthesis, but very high concentration may prove toxic, and rate of photosynthesis will go down. So another external factor is temperature. Usually an increase in temperature from 10 to 40 degrees C bring about an increase in the rate of photosynthesis. So as you will increase the temperature beyond 40 degrees C, then it will put negative effect on the function of different enzymes of C3 and C4 cycle, and which put negative effect or which can results in decrease in the rate of photosynthesis. Another important factor, external factor, is oxygen. So increase in the concentration of oxygen generally decrease in the rate of photosynthesis, and this process is known as Warburg effect. So Warburg effect generally states that if the concentration of oxygen increases, the rubisco gets affinity for it, and a process which is waste of energy carried out known as photorespiration. This is observed in C3 plant, in which glycolate acts as substrate and reduces the rate of photosynthesis. Few internal factors are also in the plants which put effect on the rate of photosynthesis. That is protoplasm factor. Here, proper hydration of protoplasm is essential for photosynthesis. Another internal factor is anatomy of leaves. So different anatomy of leaves also put effect on the rate of photosynthesis. As I already explained you in the earlier part of my presentation, that C3 plants are different leaf anatomy and C4 plants are different leaf anatomy. So by different, so how different in leaf anatomy put effect on reducing the rate of photorespiration by increasing, and this helps in regulating the rate of photosynthesis. So this is all about the chapter photosynthesis. So if you have any problem in understanding anything regarding this chapter, you can call me, you can message me. So thanks to all of you. Bye bye.